Hi, my name is Rebecca MacLeod, and I'm the Director of Technology here at CellsBin. I'm really happy to tell you today about our technology and our developing product, CB Insight. This is a platform that allows us to identify rare cells at very low levels with a focus on cancer diagnostics and disease progression tracking. We are based near New Haven, Connecticut, and we use nanophotonics, AI, and microfluidics to identify very rare cells from a background population. One application of this is in cancer diagnosis and treatment. Our product CB Insight aims to help clinicians diagnose and monitor the progression of cancer, specifically multiple myeloma. We have found that our technology can identify as few as one cell in a million, which is an order of magnitude greater than current methods and particularly important in early cancer detection. Multiple myeloma is an aggressive form of blood cancer. It can accelerate from relatively benign forms that can persist for a long period of time before becoming really aggressive and fatal. The main method of diagnosis right now is a bone marrow biopsy, which is painful and invasive and not a routine procedure. We're developing a commercial lab-developed test that will allow clinicians to diagnose multiple myeloma with a quick blood draw, as circulating tumor cells are present in the peripheral blood of patients, but at much lower levels than what are found in the bone marrow. To accomplish this, we have developed an instrument called Vega, here on the left. This instrument flows cells through a microfluidic chip while acquiring high-resolution bright field images and simultaneously fluorescent marker intensity. You can see in the photo on the right that our instrument is much smaller than the typical flow cytometer, and as it uses microfluidics, it requires very little reagent for operation. Our instrument acquires the data from the flowing cells, and the images and fluorescent intensity are analyzed in a 2D array as they flow. This allows us to flow the cells through gently without compromising throughput. The images and fluorescent signals are fed through an AI algorithm that finds cells of interest based on morphology and photonics-based fluorescent signals. The data are processed in real time, generating an accurate cell count and images of rare cells in the sample. Some of the advantages of our system are that it does not rely on manual gating, which is subjective and time-intensive. In addition, our flow does not subject the cells to shear forces, which might render them unavailable for further processing. In our first product, CB Scout, we found that we can identify as few as one cell of interest in a background of a million, which is 1 to 2 orders of magnitude of improvement over even high-end flow cytometers. We're now pursuing an IRB with Yale University Department of Hematology, where we are specifically addressing multiple myeloma, as it is a disease with complex progression, low survival rate, and as of yet, invasive diagnostics. This graph shows that when we spiked multiple myeloma cells into whole blood, we were able to quantitate those cells in a linear fashion to levels down to tens of cells in 1 milliliter of blood. These results are very promising, and we are now continuing our validation with multiple myeloma patient samples and comparing side by side with flow cytometry. We're using our technology also to track and quantitate therapeutic cells over time, as this is an area of active research as immunotherapy develops. Our next steps as we finalize our LDT are to not only identify rare cells, but to isolate them for downstream applications. Especially for rare cancers, having the ability to analyze rare viable cells at a single cell level will be very valuable for researchers to conduct analyses such as sequencing, multiomics, and even culturing. Most current isolation methods are based on binding affinity, but since our method is based on morphology and phenotypic expression, we may be able to isolate cell types that were previously unavailable or isolated inefficiently. We are very excited about this work and are awaiting the result of an NIH Phase 2 SBIR to pursue this study. Thanks so much for your attention, and I hope to see you all in Florida.